Prokaryotic translation, elongation. In this step, the translation machinery has the onus of the catalysis of peptide bond formation and also ensuring the specificity and fidelity of the translation process. That translation makes an error at the rate of 1 in 1000 amino acids that have been incorporated into the polypeptide is thanks to the fact that during this elongation step, care is taken to ensure that a correct tRNA brings the amino acid for the peptide bond formation. So let us look at the learning outcomes of this session. At the end of translation initiation, the 70S complex is bound to the initiator tRNA strategically with the A site free to bind to another amino acyl tRNA to actually begin the elongation step. Three elongation factors EFTU, EFG and EFTS help in regulating and coordinating the elongation step, which ensues the process of forming the peptide bonds between amino acids based on the sequence of codons in the mRNA. And EFTU, EFG, and EFTS are able to uh, enable the smooth flow of events right from adding the first amino acyl tRNA to the last amino acyl tRNA. The ribosome ensures that the correct initiator tRNA occupies the A site, thereby contributing to ensuring that errors are less. The ribosomes translocates along the mRNA, synthesizing the protein until it reaches the termination codons. Energy is required during this process of elongation. So let us look at what are the important components of the elongation step. Of course, what has happened in the initiation step is something that is required by the elongation. So you need the entire assembled 70S complex with the amino acyl tRNA attached to the P site and the A site and the E site being empty. This is needed. Then you have a pool of charged tRNA, okay, acylated tRNA with the cognate amino acids that can now enter the A site to continue the synthesis of polypeptides. And aiding these two are three elongation factors, EFTU, EFG, and EFTS. Now, all of these elongation factors are associated with the nucleotide GTP. And this association of the elongation factors with the nucleotide, that is with the GTP, has a regulatory role to play, as we will see with the progress of the steps in elongation. So let us look at the first step of elongation, and that is basically the entry of amino acyl tRNA to the A site. So what do we understand by that? So let us look at the fact that post the initiation or end of the initiation, you have the assembled 50S, 30S into the 70S complex. You have the complete E site, P site and the A site. And within the P site, you have the initiator tRNA, which is firmly bound to the start codon of the mRNA. And the positioning of the mRNA, etc., was all carried out in the initiation step itself. So we are left with at this position strategically that the A site is empty to take in another tRNA. So therefore, what is again important in all the elongation steps is the presence of the factor binding center at the 50S, that is a large subunit of the ribosome, and also of course the catalytic site or the active site, which is the peptidyl transfer center, which has ensconced within it the 23S rRNA that carries out the catalysis. So it is basically the 23S rRNA that acts as a ribosome and forms the peptide bond. So these two centers or these two regions in the 50S subunit are very important for the elongation step. Now, what we therefore understand is from the pool of amino acyl tRNA, you can have the amino acyl tRNA occupying the A site. Now, it is true that because all amino acyl tRNA structurally are more or less similar, you could have any of the amino acyl tRNA coming and occupying the A site in the ribosome. However, it is now the ribosome's onus to actually ensure that an incorrect amino acyl tRNA does not occupy the A site. Moreover, what has been reported is that the amino acyl tRNA do not come and occupy the A site on their own. Rather, it is the elongation factor Tu which escorts the amino acyl tRNA to the A site. So this is what we are going to look at next, is what is EFTU, that is elongation factor Tu, and how does it bring in the amino acyl tRNA to the A site. Now, looking at the elongation factor Tu itself, you can see that the elongation factor Tu can exist in two conformations. So, you can see there is the conformation a little different over here from the conformation over here. So, a monomeric protein with different domains, as you can see, okay, can take up two different conformations, and that is decided by which nucleotide it is bound to. So when a GDP, that is a guanine nucleotide diphosphate form, is bound to the EFTU, then the conformation that it takes up is this. But when it is bound to GTP, okay, then what you can see is there is a shift of this domain to forming something that can act as a cap. So you can have something bound over here. Okay. So which means that the GTP is ensuring that you have such a conformation that is having a high affinity to bind to the tRNA. So we will look at that in a minute. But what is obvious, very obvious over here is that the binding of GDP leads to its conformation being different, and the binding of EFTU with the GTP has a different conformation. And therefore, this aspect, that whether it is bound to GDP or whether it is bound to GTP and that it can have different conformations, have a regulatory connotation to that. So it is kind of like a switch on, switch off kind of system, where when GTP is bound, it has a definitive function, and when GDP is bound, it has a different function. So the understanding therefore is that an amino acyl tRNA at its 3' end, where it is acylated with the amino acid, the EFTU GTP is coming and binding to this region. So effectively what has been observed is that if EFTU has very high affinity for all amino acylated tRNAs, it can go and bind to any tRNA that is charged, and it will go and bind only to the 3' end of the tRNA when it is acylated, not when it is not acylated. So if you don't have an amino acid component over here, then it is observed that if EFTU GTP binds rarely to the so it is effectively understood that the EFTU GTP binds to a charged tRNA, and that is attributed to the fact that when it is bound to the amino acyl tRNA with the amino acid, the conformation of the EFTU GTP ensures that the amino acids that are present, the microenvironment within the EFTU, is able to interact with the acceptor arm and also the T-Cy stem. So this part, this entire part of the tRNA, this part is the acceptor arm and this part is the T-Cy stem. So this can interact both with the acceptor arm as well as the T stem. So this interaction makes the affinity with which the EFTU GTP binds very stable. And interestingly, what is to be noted is that EFTU GTP has an inherent GTPase activity, but its GTPase activity is not induced when it is in the EFTU GTP form attached to the charged tRNA. So, in this conformation, in this interaction with the charged tRNA, the GTPase activity of the EFTU is not activated, and this in turn has an important role to play in terms of ensuring specificity of translation, and this we will just look in a few minutes. Also strategically, the amino acid present at the 3' end of the tRNA is masked by the EF2-GTP. So until the EF2-GTP is attached to the amino acid is not available for forming a peptide bond. Only when the EF2-GTP is released, you will notice that the amino acid is now free to move a peptide bond. So therefore, you can see that this is another regulatory phase of EF2-GTP. Now let's consider, say for example, the EF2-GTP becomes an EF2-GTP after hydrolysis. So suppose the GTPase activity of the EF2 gets active, and because of that you have the GTP getting converted or hydrolyzed to GDP. Now when GDP is bound, we have already seen that there is a conformational change. So that conformational change has the low affinity for binding to the charged tRNA, and therefore EF2-GDP will leave the charged tRNA. So effectively what we know is that in the form of EF2-GTP, it can bind to the charged tRNA at the 3' end, but in the form of EF2-GDP, it cannot bind to the acylated 3' end of the tRNA. Therefore, both these conformations together are kind of contributing to the fact that amino-acyl tRNA is brought to the A site, but not just brought to the A site, it also helps the ribosome to ensure that a correct amino-acyl tRNA is present on the A site. So the fidelity of the mechanism is maintained also thanks to EF2-GTP, and we will see that in a while. So going back to where we were, okay, is that the A site is free, the E site is free, and the P site is occupied by the initiator tRNA with the FMET present. Now, the EF2-GTP is bringing the amino-acyl tRNA to the A site, okay, and interestingly, what is to be noted is that if the positioning of the tRNA is proper, then you would always find that the EF2-GTP will be positioned directly on the factor binding center. But this has to do with the fact that the positioning of the tRNA at the A site has to be proper. Otherwise, this will not sit within the factor binding center. It will just lie outside the factor binding center. So here when you have the tRNA coming into the A site, you have the mRNA over here. So let us look at the fact that this is the mRNA, okay, and the tRNA is brought into the A site. So what is important is the forming of the hybrid between the anticodon and the codon. So if the tRNA has the correct anticodon for the codon, then you will have a stable interaction between the tRNA and the, sorry, I forgot to put the EF2-GTP over here. So the interaction over here is going to be very stable. And this effective, stable hybrid formed between the amino-acyl tRNA and the mRNA is something that the ribosome is looking at to ensure that it is not an incorrect tRNA that is coming into the A site, but it is the correct tRNA coming into the A site. So this is one mechanism by which the ribosome is ensuring fidelity. Okay, and another interesting point to note over here is therefore, when you have the tRNA with its anticodon forming a stable interaction with the mRNA, then you would have the EF2-GTP positioned at the factor binding center. Now, this is so effective that even if there is one mispairing, that is base mispair. So you have one nucleotide of the anticodon not complementary to the codon or one of the nucleotides in the codon, then it is observed that you will not have proper positioning of the tRNA on the codon, that is on the mRNA, and you will not have this interaction proper. So when this interaction is not proper, look at the way the tRNA is not correctly positioned on the A site, and thereby you would have the EF2 GTP not positioned at the FBC. It is side tilted. So it will be outside the FBC, that is the factor binding center. So this is one important fact, okay, that the ribosome takes note of. Now, another aspect, therefore, this is the first checklist. Now it does the second checklist, or it makes use of another mechanism to find out whether the tRNA that has come in is the correct tRNA or not. So, when you have the anticodon and the codon interacting with each other, and if the anticodon is completely complementary to the codon, okay, irrespective of its third wobbling nucleotide, the third wobbling nucleotide also makes an interaction, and that interaction also contributes the stability of the final anticodon-codon binding. So if this interaction is stable, okay, or if this interaction is correct, then that forms a mini helice, with the helice having a proper minor groove and major groove formed. We know that every base pair contributes to formation of the minor groove and the major groove. So if the base pairing is not correct, it will not form a proper minor groove and a major groove. Okay. Now this fact is taken advantage of by the ribosome to look at whether a correct minor groove has been formed or not. So how is that done? So at this site where you have the A site, you know this pocket, half the pocket of the A site is part of the 30S and half the pocket is part of the 50S. And just below the pocket, or in fact the entire subunit has the 16S rRNA, different parts of 16S rRNA positioned at different places. So just below the A site at this point is present the rRNA with a conserved set of A's. There are two conserved A right below the A site over here. Okay, so I'm showing it here. So this AA, conserved A, will interact if a proper with the tRNA and the mRNA hybrid form, if and if only the minor groove formed is correct. So this 2A of the 16S rRNA to interact with the mini helice formed over here, the catch is that the minor groove has to be formed properly. So, in fact, if the minor groove is formed properly over here, then we know that the A's that are present in the 16S rRNA will also interact with this. And so, you have over here three interactions. So, it's a ternary complex that is formed. You have the tRNA interacting with the mRNA, which in turn is being interacting with the 16S rRNA. And so you can envisage that the stability is going to increase. Of this interaction, the stability is going to increase, and therefore this is the second mechanism by which the ribosome is able to ensure that an incorrect amino acid tRNA is not occupying the A site. If it is an incorrect amino acid tRNA, then you do not have proper positioning of the anticodon with the codon. You will not have probably even a single nucleotide. If it does not interact with the nucleotide in the codon, you will not have a proper minor groove formed. And if you don't get a proper minor groove, the AA of the 16S rRNA does not interact with the codon anticodon. And thereby the ribosome can ensure that this is not the correct tRNA. Now, so we have been envisaging, therefore, that if everything goes well, that is, you have a correct codon-anticodon interaction and you have the 16S rRNA interacting with that codon-anticodon, then the EF2 GTP will now be positioned at the factor binding site. So when this is nicely ensconced within the factor binding center, there will be a conformational change in the EF2 because of which you would have its GTPase getting active. Okay, and with that, therefore, what is going to happen? The inherent GTPase activity will hydrolyze its GTP into GDP. And we all know that when you have the EF2 bound to GDP, its conformation has very low affinity for the charged tRNA, and therefore it will leave the factor binding center and also leave the tRNA in the A site. So now you have the A site being occupied by, as per the two mechanisms, a correct tRNA. Okay. However, if this tRNA still is fooling the ribosome, then there is one more mechanism by which the ribosome ensures that the amino acid tRNA present in the A site is correct or not, and which is what we are going to look at now. So now we know that you have a amino acid tRNA at the A site interacting with the mRNA, interacting with the 16S rRNA. Okay. Now, what is the next step? The next step is that this amino acid that has entered the amino acid tRNA should form a peptide bond with fmet, because that is the ultimate goal of the ribosome, that is formation of the peptide bond between two amino acids. So here, as we can see, the fmet has to now be in a peptide bond with the AA. Positioning of the tRNA in the P site and the A site is set such that this amino acid is away from the catalytic site, which means that to form the peptide bond, this amino acid should come closer to the peptidal transfer center in order that the 23S rRNA present in the peptidal transfer center interacts both with the fmet as well as with the amino acid, as these two are their substrates. Correct? So, in order to have the amino acid present close to the peptidal transferase center, this tRNA will have to twist within the A site, reposition to bring the amino acid close to fmet. And that is what is happening by twisting completely. So, it's almost a 180-degree turn that it makes, and this 180-degree turn that the tRNA makes within the A site is what is called as accommodation. So it accommodates itself within the A site to ensure that the amino acid that it is carrying is close to the active site. It's close to the active catalytic site. Now, definitely when you have this anticodon arm twisted in the A site, there is going to be a strain that is created between the codon and the anticodon, because you can imagine that there is a twist. Because there is a twist, there will be a strain created. Now, here is where the catch is. If the interaction between the codon and the anticodon is not stable, then due to this twist, you can have the bonds breaking. Okay. However, if it is the correct codon and anticodon, then the hydrogen bond will hold together still to sustain the strain that is formed. So, this sustenance of the strain will decide whether it is the correct amino acid tRNA or not. And therefore, this accommodation is sustained if the base pairing is correct. And this forms the third checkpoint of the ribosome to ensure that it is not allowing an incorrect amino acid tRNA in the A site. Okay, having done that, next is the formation of the peptide bond between fmet and the amino acid. So the incoming amino acid, its NH2 group, is able to cleave the acyl bond between fmet and the tRNA, and that acyl bond is a high energy bond. So the energy released due to breaking of that acyl bond is used by the 23S rRNA to form the peptide bond with the amino acid that has come in. Okay, so interesting to note over here is that the initiator tRNA now is uncharged. It does not carry the fmet any longer, because the fmet has now shifted to this amino acid tRNA. Why? Because it is in a peptide bond with the amino acid that is attached to the amino acid tRNA at the A site. So the tRNA at the A site is now not carrying one amino acid, but it is carrying a dipeptide. So basically, you will find that even if there is a growing peptide over here, that growing peptide will be shifted to the amino acid tRNA at the A site after the peptide bond formation. So this is significant. Now we come to what happens next. Okay, so the next step that is going to happen is something called translocation. So let us just basically get an overview of what translocation is. And having got the overview of what translocation is, we will look at how this is carried out. So once you have the peptide bond formed between the F-met and amino acid, in fact, the peptide bond is the first step that is contributing to translocation. So what happens is, once you have the peptide bond formed between F-met and the amino acid of this tRNA, you will observe that the ribosome moves one place in translocation, and you will have effectively this tRNA shifting to the E site and the tRNA at the amino acid site moving to the P site. So with the ribosome moving this way, you would have these two shifting this way. So therefore that is what is translocation effectively. And also important to notice that as it moves from here to here, you can understand that this was bound to AUG. So because it was bound to AUG and the interaction being strong enough, the AUG is also going to move here, and this being bound to AAA, the AAA will also bound to the P site. So the tRNA at the P site shifts to E site, the tRNA at the A site shifts to the P site, okay? And as the interaction of the tRNA with the mRNA is stable, the mRNA is also shifting. So everything is shifting by one codon, one triplet codon, if you can note, okay? So that is the very interesting fact about translocation. And of course, strategically, therefore, now the P site carries the amino acid tRNA with the peptide. And that's why this site is called as the peptidal site. And of course, you have the A site again free for another amino acid tRNA to come and bind, corresponding to the next adjacent codon in the mRNA. And you have the tRNA from the E site moving away. That is why it is called as the exit site. So now let us look at how this all happens. So for all of what we discussed to happen, you have over here another elongation factor G, that helps in the process. So observantly, like I told you, the peptide bond formation itself is responsible for beginning the translocation. So you can see that with the formation of the peptide bond, the tRNA on the P side tilts to the E side, the upper acceptor arm side tilts to the E side, and the tRNA that is present in the A side is also partially tilted towards the P side. So the 3' end of both the tRNA, as you can observe, is tilted towards the other side. So you may over here basically claim that translocation in the larger subunit is preceding the translocation in the smaller subunit, because the anticodon stem of the tRNA is still positioned at the site that it originally was. That is, this fellow is still bound to the A site strongly, and this is also still bound to the P site strongly. But their upper acceptor arms have tilted towards the E site and the P site, indicating that that is where the tRNA is going to shift with translocation. Now next what happens is that you have the EFG that is going to help the anticodon part of the tRNA shift to the E site and the P site respectively. Now EFG is also bound to a GTP, so it is EFG GTP. This also has an inherent GTPase activity, and like EFTU, when it is bound to GTP, it has a different conformation, and when it is hydrolyzed and it is bound to GDP, then it has a very different conformation. So that is what we are going to observe again. So this also has the same aspect as EFTU, that because of the conformational changes, there is a regulatory effect. So over here, what is important to notice that this EFG GTP is able to bind to the factor binding center of the ribosome. So therefore, it comes and binds to the ribosome at the factor binding center. Once it binds to the factor binding center, because of its interaction with the large subunit and a conformational change within it, its GTPase activity is initiated, and because of that, what you would have is this GTP is converted to GDP. But what is interesting is, look at the way it has changed its conformation. One important thing to be noted over here is that the conformation that it takes up when it is bound to GDP is very similar to a charged tRNA. Okay, and therefore such a phenomenon where one molecule is mimicking the other molecule is called as molecular mimicry, and this molecular mimicry plays a very important role, especially in transcription and translation. As known in transcription, the sigma subunit of the RNA polymerase, some domains of it seem to mimic the DNA and the RNA, and here you can see that the elongation factor, when it is associated with the GDP, it is forming a structure which is very similar to the charged tRNA. So therefore, now what happens is, when it is converted to the GDP, look at the fact that it is trying to accommodate itself within the A site. And if it is trying to accommodate, it is going to push the tRNA out from the A site. And that is exactly what happens. Although the intricate details of this is not known, but this is exactly what happens. That is, the EFG GDP is able to literally push the two tRNA from their existing sites to the next sites. And as mentioned earlier, along with the tRNA, the mRNA is also shifting. Okay, so now what you have is, because the tRNA has shifted from this position to this position, you can invariably note that the 30S sees some conformational changes. And with those conformational changes, the affinity of the binding of EFG GDP to the A site decreases and it leaves the ribosome. So what does that mean? It means that now you have again the A site empty after translocation, and you can have another if EF2 bringing in another amino acid tRNA. The ribosome ensures that it is the correct tRNA, binds to the correct codon, binds to the codon, and ensures that it is positioned well again to form a peptide bond with the dipeptide that is already there. So therefore, this dipeptide will then shift to the amino acid, that tRNA that comes to this position. And likewise, you will have the second cycle of elongation. Till here, it was the first cycle of elongation. Now with the next EF2 GTP bringing the next amino acid tRNA, the next cycle of elongation begins. And this will continue till the ribosome reaches the termination codon. What is important to note again is that as the conformation is changing within the 30S, you have the EFG GDP moving out of the ribosome, but what you also observe is that the mRNA goes farther away from the E side, and there being a conformational change, the affinity with which the tRNA binds to the codon in the mRNA is less in the E site, and therefore the tRNA that is present in the E site also exits. So you again come to a state strategically where only the P site is occupied and you have the A site and the E site free. So just looking at the structure of EF2 GTP, so you can see this is the EF2 GTP bound to the tRNA. When you look closely at the structure of EFG, you see a similar structure absolutely. These are the domains of the EFG which form a very similar structure as the tRNA EF2 GTP complex. So this is what is called as the molecular mimicry. Now, what is the role of the next elongation factor, that is EFTS? So EFTS is notable in restoring the EF2 with a GTP again. So primarily, you know that when EF2 is released from the ribosome, it goes out as EF2 GDP. Also, it is interesting to note that EF2 GDP cannot bind to a charged tRNA. So the EF2 GDP has to get converted to a EF2 GTP to again bind to a charged tRNA. And when it binds to a charged tRNA, it can again bring the charged tRNA to the ribosome because it can associate with the factor binding center. So what happens is when EF2 GDP is formed, it can interact with EFTS. So what forms is an EF2 EFTS complex. With the formation of the EF2 EFTS complex, a GTP can again associate with the EF2. And on binding of the GTP to the EF2, thanks to the conformation that the EFTS was able to bring about in EF2, in order for the GTP to come and bind, the EFTS can remain associate and help the EF2 GTP to bind to a charged tRNA. So this is one path that it can take. The second path that it can take is that the complex is formed, the GTP is bound to the EF2 GTP, and the EFTS then is released. Okay, so these two pathways are basically ensuring that the EF2 is again associating with a GTP, and that helps it to interact with the charged tRNA again. So it is basically EFTS has a role only to ensure that the EF2 GDP is back again charged with GTP and the GDP is released. Now, the question that comes over here is, what about EFG? EFG does not need the help of EFTS in restoring its GTP because EFG as a molecule has a very high affinity for GTP. So as soon as it becomes EFG GDP, it spontaneously releases the GDP and it preferably takes up a GTP again to become EFG GTP. Okay, so the charging of the EFG with the GTP is done by itself because it has a higher affinity to bind to GTP. Now let us just look at the energetics of the elongation process. So as you can see, the EF2 GTP is hydrolyzed when the ribosome has ensured that due to the correct interaction that the amino acyl tRNA makes with the mRNA, you have the EF2 GTP positioned at the factor binding center, and when it is positioned at the factor binding center, its GTPase activity is ensued, and because of that it gets converted to GDP. So here you have use of one energy molecule, and that energy molecule is GTP. So the hydrolysis of that GTP gives rise to or utilizes an energy molecule over here. The next is the peptide bond formation itself. But here no exogenous nucleotide is used. Why? Because the high energy is coming from the acyl bond. And from where did the acyl bond come? The acyl bond came from the fact that the tRNA, when charged with an amino acid, the amino acyl tRNA synthetase uses an ATP to form that acyl bond between the amino acid and the tRNA. And that acyl bond is storing the energy of that ATP used. Okay, and so the energy of that ATP is being used in the peptide bond formation. So that is another ATP molecule that is used. Then of course, during translocation, for the translocation process, the EFG GTP hydrolysis takes place, and that is the third energy molecule that is used. So therefore, for every peptide bond formation, three energy molecules are being used. Two are GTP and one is ATP. ATP indirectly because that ATP was added when the tRNA was charged in the first place. Okay, so these three molecules are important for peptide bond formation. Therefore, now let us make the conclusions. The EF2 GTP brings a charged tRNA to the A site, and following specific mechanisms, The ribosome ensures that the correct charged tRNA is in the A site. What are the mechanisms? One, ensuring a proper codon-anticodon binding. Second, ensuring codon, anticodon and ribosomal RNA interaction. And of course, the EFTU GTP getting hydrolyzed to EFTU GDP. That will not happen unless you have the correct codon-anticodon interaction. And the third, of course, is accommodation. With two charged tRNA on the P side and the A side, positioned such that the substrates of the peptidyl transferase are at the peptidyl transfer center, the peptidyl transferase, which is the 23S rRNA, forms the peptide bond between the substrate on the P side and that in the A side. Thereafter, the uncharged tRNA at the P side and the tRNA carrying the growing peptide on the A side are translocated by the EFG such that only tRNA with peptide is present on the P side, leaving the E and the A side free for the next peptide bond formation cycle. The EFTU GDP gains back its GTP by complexing with EFTS, while EFG GDP spontaneously takes up GTP as it has greater affinity for GTP. Thus, elongation is a sequential and smooth flow of regulated events that ensure that the correct tRNA binds to the ribosome, which then forms the peptide bonds to enable formation of the polypeptide as per the codons in the mRNA. Thank you.